Good day, dear student. Today we will discuss another important group of biomedical tests used in medical bacteriology, which is called MVAC test. As a medical lab student, we should understand that biochemical tests are essential for identification of bacteria according to the metabolic activity. Now, we will explain what we mean by the MVAC test. Now, what we mean by the MVAC test is a group of 4 biochemical tests used for identifying members of the Enterobacteriaceae family. These tests include I. indole, M. methylred, V. vojas proskauer, C. citrate. And these tests are essential and useful to differentiate between Escherichia coli and Klebsiella pneumoniae, which appear similar in culture media. Now we begin with the first test, indole test. The indole test. The aim of this test is to detect the ability of bacteria to produce the enzyme tryptophanase. This enzyme breaks down tryptophan into the indole, ammonia, and pyruvic acid. As I mentioned, MVAC test used to determine the Enterobacteriaceae family. Some membrane in this family produce the tryptophanase. This enzyme breaks down the tryptophan into the indole. We can detect the bacteria that produce indole by using the chemical reagent, which is called Cobas reagent. When we add this reagent to the colony of the bacteria, if the bacteria have the ability to produce the tryptophanase and break down the tryptophan into the media, produce the indole and give the red color by add this reagent. Now, how to perform this test in a lab, I discuss in the next slide. The procedure of the indole test. How to perform this test in a lab. First, we inoculate the bacteria in a peptone water broth. After that, we incubate it at 37 for 24 to 48 hours. After incubation period, we add the reagent, Cobas reagent. The positive result appear as red ring like E. coli. The negative result, yellow ring at the top like Klebsiella pneumoniae. Why this happen? This media, peptone water broth, contain the tryptophan. If the bacteria have the tryptophanase, it breaks down the tryptophan and produce indole. After add the reagent, we indicate it as a red ring at the top. That means the bacteria have ability to produce the indole like E. coli. And that's all about the indole test. We move to the next test, which is called methylred test. Methylred test. This test is used to detect if the bacteria have the ability to ferment the glucose completely and produce large quantity of acidity, such as acetic acid, lactic acid, formic acid, which lower the pH of the media below 4.5. And this test is performed in a glucose phosphate peptone water, which is also called methylred vojas proskauer broth. And the indicator used is methylred. When this indicator is added to the media, the red color indicates there are sufficient acidity have been produced by the bacteria and the test is positive, as we can see in E. coli. If media remain yellow, that means the organism has not produced enough acidity and the test is negative, as in Klebsiella. The procedure of the test. We inoculated the test bacteria in a broth media. As I mentioned, the broth is also called methylred vojas proskauer broth. Why we called? Because it's used in a methylred test and vojas proskauer test. After that, we incubated at 37 Celsius for 24 hours to 48 hours. After incubation time, we add the pH indicator methylred 5 drop and mix. The red color, it indicates strong acid production, while the yellow color, it indicates the negative result. And the positive result example, E. coli, and the negative result example, Klebsiella pneumoniae. Now, Voges-Proskauer test is used to detect the ability of the bacteria to produce the acetone from fermentation of the glucose. This fermentation is incompletely lead to production of the acetone. How detect these bacteria? Use the media like in methyl red that are called glucose phosphate Pupton water or methyl red Voges-Proskauer broth, and two reagent A and B, 40% of the KOH, and reagent B, 5% of the alpha naphthol. We add the first reagent A, to the media. This reagent oxidizes the acetone that are produced by bacteria to diacetyl, and then we add the second reagent B, alpha naphthol, that react with this product and produce the red color. The procedure of this test: we inoculate bacteria in a broth, methyl red Voges-Proskauer broth, and incubate it at 37 for 24 to 48 hours. After incubation time, we transfer 2.5 milliliters of the broth that contain the bacteria and inoculate it in another tube. Add 6 drops of the reagent A, then add 2 drops of reagent B. Mix gently and wait for 10 to 15 minutes. The positive result appear in a pink or red color, and the negative result, no color change. The media remain yellow. Example of the positive result, Klebsiella pneumoniae. Example of the negative result, E. coli. That is mean Klebsiella pneumoniae ferment the glucose incompletely and produce the acetone from this fermentation. Now we will move to the citrate test. This test used to detect ability of the bacteria whether can use the citrate as only source of carbon and energy. Bacteria that have enzyme citrate permease can transport citrate into the cell and use for growth. This enzyme helps bacteria to use a citrate into the cell and use for growth. And the medium used is semen citrate agar that contain bromothymol blue as pH indicator. If bacteria can use citrate, alkaline products are formed and the media change from green to blue color. And the test is performed on a slant agar because the oxygen is required for citrate utilization and the slant provides large surface exposed to air. The procedure is very simple. We inoculate bacteria in the semen citrate agar that contain bromothymol blue indicator by stamping and streaking method. On slant agar, we use stamping and streaking method. Incubated at 37 Celsius for 24 to 48 hours. And the positive result appear as a blue color and they are growth on a surface of the slant, like in Klebsiella pneumoniae, that indicate the bacteria use the citrate and produce the alkaline pH, while the negative result is indicated by green color and no growth in slant, like in E. coli. So we concluded that Klebsiella pneumoniae produced the blue color, indicated the alkaline pH and citrate utilization. In this table, you can show the E. coli is positive for indole and methyl red negative for Voges-Proskauer citrate, while Klebsiella is negative for indole and methyl red positive for Voges-Proskauer and citrate. Salmonella typhi negative for indole, positive for methyl red, negative for Voges-Proskauer and positive for citrate. Shigella dysentery is negative for indole, positive for methylred, negative for Voges Proskauer, and negative for citrate. This indicates that Invictus is very important for identification the species of the Enterobacteriaceae and differentiate between them. And we concluded that Invictus is a group of 4 biochemical tests used to differentiate between Enterobacteriaceae family. We are looking for a specific color change. For example, a pink ring in indole test, it indicates the positive result. In methyl red, the red broth indicates the positive result. In Voges Proskauer, the red surface indicates the positive result. In citrate, the blue color it indicates blue slant, it indicates the positive result. We checking which tests are positive and which are negative, and these are useful for differentiation, especially between the coliform bacteria like E. coli and Klebsiella pneumoniae. And this video link, watch this video to see how the tests are done step by step in a lab.